Hi, I'm Annaliese Mason from the University of Bonn. Thank you for the opportunity to give this talk. I'm the new Professor for Plant Breeding. So, world agriculture is not very diverse. Wheat, rice and maize together comprise more than 50% of all plant food worldwide, while just 9 crops supply more than 75%. In many species, diversity is limited, and this also limits possible breeding gains. For instance, there's little natural insect resistance in rapeseed, and more than 95% of bananas grown today are a single clonal cultivar, the Cavendish. Better use of diversity in cropping systems could offer potential benefits to human nutrition, agricultural ecosystems, and in building climate resilience. This is not so new per se, but the core research theme of my group is polyploidy and hybridization for crop improvement, which is a special way of introducing diversity. So what is polyploidy and hybridization? Or should we start with what is a hybrid? Lions and tigers sometimes hybridize. So this means that we get an interspecies hybrid between lions and tigers occasionally. What's the most commonly known example of this in animals is actually when we cross a horse by a donkey to get a mule. So mules are sterile, they're unable to make baby mules. And one of the reasons for this is that horses and donkeys just have different sets of chromosomes. So we refer to the set of chromosomes present in an organism, often with a letter. This is the genome. So when horses and donkeys hybridize, we get mules, but these poor mules have 63 chromosomes, which is an unbalanced genome complement, so they can't actually reproduce. Hybrid species, on the other hand, are also quite common, and these are very common in plants particularly, which tend to be a bit more flexible. And if we start with species 1 here and species 2, so we're going to assume that species 1 just has 2 chromosomes, like 1 pair, which is the A genome. Species 2 has another set of chromosomes, a different set of genetic information, it's the B genome. And if these can actually also hybridize to produce a hybrid species with a complete set of genetic information or all chromosomes present in the parent species. So this is a hybrid species or an allopolyploid. So we have our interspecies hybrids, interspecific hybrids like the mule, and we have our hybrid species like allopolyploids. Polyploidy actually just means having more than the diploid chromosome number. So humans are diploids, meaning 2. We just have 1 set of chromosomes from our mum, 1 set from our dad. So that's like every chromosome is paired. We can also have triploids, so that's 3 copies of each chromosome. And we can have different flavors of polyploids, so tetraploid or higher. Can come in allopolyploidy, which means that there are 2 or more different species or different origins of the chromosome sets, and autopolyploidy, which means that chromosome doubling has happened within a species. Probably already asking, why should we care about hybrids and polyploids? Well, we probably ate some already today. So interspecies hybrids include apples and citrus fruits, which are hybrids between 2 or more species. And we utilize the fact that triploids are sterile, because they have 3 copies of each chromosome, to make our seedless fruits, watermelon and banana. Also polyploid or hybrid are bread wheat, oats, wheat, rapeseed, kiwi fruit, sugarcane, strawberry, and many more. So these are potentially very interesting for our agricultural plants. This is also a very common process in ancestral evolution of the seed plants and angiosperms. So this is very old now, this is from 2011. This was all of the currently available genome sequences. So down the side we have species such as papaya, poplar, cucumber, grape, rice and sorghum, for instance. And what we're looking at here is a phylogenetic tree. And we can identify polyploidy events have occurred right at the base of the seed plant diversification and also the angiosperm diversification, as well as several more that have occurred more recently. And these are thought to actually have improved the diversification and speciation rates of these plant species and these plant groups. So all seed plants have at least 1 polyploidy event in their past. Also frequently polyploid, fish, amphibians, fungi, insects, actually everything except mammals and birds, which is why you probably haven't heard about it so much. But why are polyploids so successful? There's actually a number of possible reasons for this. The first one is hybrid vigour. It's one of my favorites, so I put it first, but actually we don't really know what factors contribute so much. But this is when we have 2 inbred parents and then the hybrid progeny between them who have different genetic variants show increased vigour. So it's like the opposite of inbreeding depression, if you've heard of that. Then greater environmental adaptation. We're getting increasing evidence for this in a number of species, recently in strawberry. So that the strawberry that you eat is an octoploid, but the little wild strawberries the diploids, Frigeria vesca, the ones you see in the forests in Germany. And the adaptation range for the bigger polyploids is much higher. Polyploids often also have larger cell and organ sizes, so this can potentially be advantageous in some respects, also for our crops. And importantly, the redundancy of gene copies allows for novel variation to arise without impacting gene function. So what this means is that normally you can't make too many changes in a gene because you need that genetic information for the viability and fertility of the organism. But if you suddenly have 2 copies of the gene, then this means that one of them can take on new functions without impacting the viability and fertility. So this allows for a redundancy and then hence for novel variants to form. So also this is linked to greater adaptive success. So one of the main research focuses of my group is to see if we can synthesize new stable polyploid and hybrid crop species, which may have some agricultural benefits. We work mostly in the used Triangle of Brassica species, which includes Brassica rapa, which is Chinese cabbage and turnip, Brassica nigra, which is black mustard, and Brassica oleracea, which is the European cabbages, kale, broccoli, cauliflower, etc. So these are quite closely related species, and they provide a variety of vegetable and oilseed crop types. These are the diploids. They have the A, B, and C genomes, which are 10, 8, and 9 chromosomes respectively. And then historically, they've come together to make our allotetraploid species, our hybrid species, Brassica junctia, which is Indian mustard, which has, or leaf mustard, which has an A and a B genome, Brassica carinata, or Ethiopian mustard, which has a B and a C genome, and Brassica napus, which is rapeseed or canola, which has an A and a C genome. And my group are interested to see if we can also artificially produce allohexaploids, which have an A, B, and a C genome. These don't actually exist naturally, but we've so far had quite a lot of success in making different hybrid combinations, for example, between napus and nigra, carinata and rapa, junctia and oleracea, and napus bicarinata by junctia. So we hope that these, in future, we can improve the viability of these as a crop. So for this, we need to look at improving the agronomic traits, making sure we're building on a lot of genetic diversity, and also improve the meiotic stability, which is another thing which is often a problem with newly formed polyploids. Another project is related to tetraploid turnips towards autopolyploid brassica crops. This is quite interesting, I think, because it turns out we've had the ability to double chromosome number in plants for a long time. So at least since the 1950s or 60s, there's been experiments in just doubling the chromosome number of various plants. Because we have often larger organ and cell size, this is potentially valuable for vegetable types. So before the 1980s, a number of commercial tetraploid turnips, kales, and radishes were produced. And these commercial cultivars have putatively already overcome a lot of the barriers we think are in place for normal polyploid establishment. So this is potentially interesting to see actually how this process works so that we can apply this for development of new polyploid crops. We're also interested in if tetraploid turnips show increased hybrid vigour over the diploids and under what conditions. The reason for this is I think this might be useful for improving our hybrid breeding schemes. So normally in plant breeding, we develop crop cultivars which are hybrid crops. There's usually within one species. So we're talking within a diploid, for instance, we have 2 parent inbred lines. So we'll say here that they have these chromosome pairs. So remember, one from your dad, one from your mum, AA and BB. These chromosomes are homologous. They're the same chromosome effectively in terms of the genes on the chromosome, but the genes have different variations. And so this is like every chromosome might have a gene for, in humans, eye color in the same location, but you might get a different gene variant like blue or brown from your mummy or dad. So hypothetically, these interactions between the 2 gene variants is what causes our hybrid vigour effect, which we call heterosis. So having 2 very different parents can give you this hybrid vigour effect. The question we have then is, do tetraploids with 4 parents have increased hybrid vigour? So we don't really know, but if we make inbred lines with A, B, C, and D, we now have 4 chromosomes in each genome. So when we make our F1 hybrids, they each have like 2 sets of genomes, and hypothetically, we should be able to construct pretty much these double hybrids, which have up to 4 different genetic variants at each locus. So does this make a difference in heterosis? We have yet to find out. Previous attempts to test this hypothesis were a bit confounded because of these meiosis issues, but I think now we have the technology and expertise from other work in my group to fully answer this question. In future, we should be also looking a bit more at this hybrid vigour effect, because clonal reproduction of F1 hybrids has just been achieved in rice via genome editing. So normally it takes quite a lot of effort to develop hybrid lines. You have to make the seed new in every generation. But if we can make our hybrids reproduce themselves without losing this hybrid vigour effect, and this is a significant advantage, and may also overcome these meiotic problems we have with the polyploids. Polyploids should have greater potential in terms of yield heterosis and adaptation to environmental stress. There's increasing evidence for this. So this is potentially an area where we want to focus more in future in breeding. So I would propose that induced polyploidy and subsequent double hybrid production could be a major leap forward for plant breeding and agricultural crops. The relationship here, I'm going to very briefly describe how plant breeding works. So normally we have a germplasm set. This is the collection of diversity of like different plants and cultivars within a species. And then over time, we select the best ones to become our like crop type. So we have all of the wild relatives, and then we pick some which have like the yield characteristics we want. And then from these, we develop our elite crop cultivars, usually through inbreeding. So we have genetically very similar crop cultivars. And this works quite well, actually. This is not a bad process in any way, but the problem is it just takes an extremely long amount of time. So we're talking many, many generations. A lot of our domesticated crop species have hundreds of years behind them already. So then if we start again with de novo domestication, so finding new species to domesticate, if we look at novel hybrids and polyploids, if we look at wild relatives, a lot of breeding companies don't really want anything to do with this because of the time it takes to move from this introduction of the germplasm into the gene pool into getting a new elite crop cultivar out of it. However, I think that with improvements in high throughput phenotyping and genotyping technologies, we should be able to improve selection efficiency. So we should hypothetically be able to shortcut our way from these individual germplasm, like this diverse germplasm set, to make diverse resilient crops. So to better utilize genetic diversity present in the wild relatives for our elite crop types. Okay, I would love to talk to anybody about this afterwards who's interested. So as an overview, I think in Brassica and many other crop species, there's a lot of opportunities to exploit polyploidy and hybridization in crop wild relatives for improvement in breeding. And we would like to look at the possibility of improving selection efficiency through developing genotyping and phenotyping technologies to better utilize this genetic diversity for crop improvement. So I'd like to acknowledge my funders and research collaborating groups in China, India, and Australia, and also my current research lab who've just moved to the University of Bonn. So thank you very much.